Now look at this.
What's up, baby biopenguins? Today we're going to do topic 4.3 on signal transduction. So this topic's a little short if you couldn't tell by the little intro. So the first step involves a ligand binding to a receptor. When that happens, there's going to be a conformational shape change. Of course, when you bind something to a protein, it changes shape. And that then leads us down this whole process of transduction. So we're going to see a lot of these intermediates going to change shape. These relay molecules will get phosphorylated. We'll have secondary messengers, all this different type of stuff happening until we end up with some type of response. So today we're going to look at specific examples of this. So in this case, we have a growth factor, and the growth factor is binding to, it looks like a tyrosine kinase receptor. And that would then go through a phosphorylation cascade. And you can see there's all these little intermediates in here. So this is going to act as a transcription factor. It's going to turn on these certain genes. And by turning on these genes, it's going to cause certain proteins to be synthesized, which is going to cause the result of the cells going to grow, which is one of the responses that we saw before. So epinephrine, we know, is for fight or flight. So when we look at this pathway, we're going to kind of keep that in mind, that this is the signaling molecule to allow you to fight or run away from something. So the epinephrine is going to bind to some type of G protein coupled receptor that activates adenylyl cyclase. Adenylyl cyclase converts ATP into cyclic AMP. That cyclic AMP acts as a secondary messenger that's then going to turn on, I believe it's glycogen phosphorylase. That's going to convert the glycogen, which is our stored glucose, into glucose. So it breaks down that glycogen into glucose. So now I have plenty of glucose in my blood that the cells can now take up this glucose and it can now synthesize ATP so we can fight whatever it is that's problematic. So look at your hand for a second. Notice you got all these spaces in between your fingers, okay? Why do you have the spaces in between your fingers? Because while you were in utero, your cells went through apoptosis. That's programmed cell death. So there was this big mass of cells here, and then the cells that were in between your fingers died, and thus were no longer there. So these cells receive a signal. It's kind of a death signaling molecule. It's going to bind to the receptor that then leads into a transduction response that's going to activate the CED3 and CED4. Not important to know those, but what is important is that these things called proteases and nucleases are activated. Proteases are going to break down proteins, and nucleases are going to break down nucleic acids. And so what's happening is the cell is going to deteriorate from the inside out. It forms these little bleeps, and then it gets engulfed by macrophage. It gets engulfed by one of your white blood cells that's supposed to kill things. Remember, doing this for every single topic, so hit that follow button, and AP Biopenguins just success. Bye, y'all.